Well, let me first thank the TBA and everyone here for all of your support throughout my program. And today I'll be talking to you about what I've been doing over the past summer, just a little bit about what I've been doing. But for those of you that don't know who I am or wasn't here last year, I'm going to fill you in on some of the previous work that I did. And some of you might be familiar with this already, but it's okay, it'll be a review. But I've done this work where I've worked with beekeepers in urban areas, and we've collected their pollen and also the nectar. And this was a two-part project. The first part was to analyze the pollen that the honeybees were collecting from in urban and suburban environments. And the second part was to actually look at the residues, do an agrochemical analysis to see if urban bees are being exposed to different chemicals and insecticides that might be harmful to them. So we did this. This is a whole national study. We have a lot of collaborators from Florida, Michigan, California, and Texas. And we want this to be an urban study, so we first did a GIS analysis. We analyzed the landscapes. So for us, we did Austin, Texas, and College Station. And we also did Florida, Detroit, and the Bay Area in California. And I've been compiling a lot of data. And this is just an example of one of the supplementary tables that we're going to have. But essentially, we have, let's see if this works. We have the family, the genus, how frequent it is, exactly the number of counts we found, the relative abundance, and what type of plant it is, whether it's a shrub, herb, or tree. So we broke this down a lot. I would love to go into this in more detail, but this will not be the focus of my talk today. But just to give you a little bit more, this is a Shannon Diversity Index. All this is saying is, measuring is how diverse each one of these sites are on average in each state and each season. So this is just a measure of diversity. So the higher the bars, the more diverse the particular season and state is. And then we also did some residue screening. So we sent out pollen nectar to the USDA to analyze these samples. And one particular compound I'm going to talk about is imidacloprid. This is a systemic insecticide, neonicotinoid. A lot of research has shown that it's not very good for bees. And we looked at different compounds of imidacloprid. And this is just our frequency of detection and infographic. We have very, very low levels of detection of imidacloprid in our pollen. So that's great news. This is at least for Austin area beekeepers. And then we have 20%, 4.7, and 5% in the other states. But yeah, we detected them. But how about quantifying them? How much imidacloprid is actually there? So these are our imidacloprid levels in each one of the states. Even though we did have a very low amount of detection in Texas, the ones that we did detect have very low amounts of imidacloprid. So, and this line over here is a colony level no observable effect concentration. So all of these values are well below that. And I think this really suggests that at least urban bees are relatively safe from this particular neonicotinoid. But with that, naturally, the question for me after doing this experiment was to look at whether or not honeybees have preferences for food. A lot of beekeepers are actually asking and talking to me about, so we see all these bees collecting all these different types of plants, but are they able to have a choice? Do they have preferences like us? Some of us might prefer one food over the other. For example, this is a breakdown of our preferences by state. I won't say what mine is, but and we also have foods that we don't like. So what I'm really trying to figure out is whether or not honeybees have this preference of food, of what they like and don't like as well. So some research has already been done, but there is still a lot more to be learned about nutrition ecology. In 2003, Cook et al., actually an Aggie, found that honeybees like overall amino acid, pollen with higher amino acids. A recent study published that they prefer a complementary diet over the other. So let's say that a bee had a diet with a lot of vitamin A. Well, the next diet, they're going to compensate for that and go for vitamin C instead to kind of balance it out. And then bees also have preferences for their salts and their minerals in water as well, from what we've seen. So the bees do have some kind of preferences, but what exactly the preferences are, we're not sure. And for what I'm doing, I'm specifically focusing on pollen. and that's because it's the main source of protein for them, and it's essential for brood rearing and developing the glands, the hypopharyngeal glands in the workers. And really, the colony growth and survival really depends on the income of the pollen going into a colony. So on average, a 10-frame hive, which consists around 10 to 15,000 workers, around 13 to 18 kilograms a year. And one way scientists use to measure what the optimal balance is using the geometric framework for nutrition. So this is such a 3D space where you can measure what is the optimal ratio of protein to carbohydrates or fatty acids or vitamins. So what this graph is showing is this is one diet where it's very high in protein, low in carbohydrates. This orange line right here is a diet that's very high in carbohydrates but low in protein. And this is a hypothetical example. And this red dot over here is an optimal intake target for an insect's like perfect growth and development. Okay, so what an organism, an individual would do if giving these two diets is they would actually balance their diet in order to reach this optimal intake target. So with that being said, there has been a few studies that's said that bees like a higher protein diet. For example, some studies have shown that high protein diet leads to better hemolymph proteins, better developed ovaries, and longer survival when stressed with pathogens or stressors such as temperature. On the other hand, other studies have found that low protein diet is actually beneficial. Some found that adults survived longer when bees were fed a lower protein and carbohydrate diet, so they had more carbohydrate, more sugar than pollen, for example. And then other studies have even found that too much protein can be fatal to both ants and bees. And even for us, to be honest, just the past year, just a few months ago, there was a news article about a bodybuilder preparing for a competition, and she had some kind of genetic disorder, but it was a factor. Too much protein was a factor in her passing away in preparation for this competition. So the research objective is to determine honeybee colony level preferences between nutritionally distinctive diets. And these are our hypotheses, whether they will forage on, or whether they have no preferences, or whether they do have preferences. So we did our study at the A&M Riverside, our now Relish campus, over the past summer. And we focused on two particular pollen types, the first of which is oilseed rape. This is brassica. And this is the third largest source of vegetable oil in the world. So this image that you see with this is an entire canola field in China, where the landscape is just, it's gold because of all the canola that's being raised out there. The next one we're looking at is that wild rose pollen. And this is an ornamental plant that's red with flowers, and it's commonly used for consumption, perfume, and flowers. So we first wanted to know what the ratios of protein carbohydrates are. So we did an assay where we took some pollen and we determined, used a Bradford assay to determine protein content. And then we used a phenysulfuric acid assay to look at the carbohydrate content. And what we essentially found is that our carbohydrates are the same, which really works out for us. We were really lucky that it was the same. But the protein content was different between these two pollen types. So the red one here is rose pollen, and then the blue one here is brassica pollen. So our rose pollen had twice the amount of protein than brassica pollen. So this already paves the way for a really cool experiment to look at whether or not they do have preference for the protein, even though the carbohydrates are the same. So we set up nucleus colonies from sister queens, and we fed them sugar and water at lib, so they had an unlimited amount, and they were kept in these cages, in these tents, and they were starved for up to a week or until all their pollen was exhausted inside of the colonies. And we did that because a previous study found that whatever a bee colony had before, they can complement their diet, so we didn't want to have that influence our results. We measured out tons of pollen, and we placed them out on a dish every day for 5 consecutive days, and we closely monitored them, and we made sure that none of them went empty. And this is our experimental setup. We had 2 groups, and then the first one had 2-to-1 distribution of rose and brassica, and the second group had a 2-to-1 distribution of brassica to rose. And we really wanted to look at the preferences and whether or not the bees are not indiscriminately foraging for pollen in an equal amount. We also did set up cameras and did some observational studies. So we took, we set a time lapse for a picture every 60 seconds. And this information should overlap with what the amount of pollen consumed. So this is just the total pollen consumed. Does anyone have any guesses whether they like high or low protein pollen before I move on to the next figure? If whoever thinks they like high protein, let's raise their hand. All right, who thinks they like low protein? All right, I think we're about to be a little surprised. So this is just a summary image of what the total overall pollen consumed. And they significantly preferred the lower protein brassica pollen over the rose pollen. And when we break it down by day, initially when they first started off, they weren't too sure. I mean, that's what it shows in their observations. If you break it down in time, the earlier time period will show that they were kind of deciding, they're learning what's going on. But as they really figured out what pollens contain, which petri dishes contain what pollen, they really preferred the brassica pollen over the rose pollen. And these letters, these different letters mean that it's significantly different from one another. So I think this is really interesting, and I actually just flew back from a conference yesterday in Denver. So we just got back in late last night, and I was talking to a lot of other people studying this type of effect, and I think that there is a general consensus that too much protein is actually fatal for bees, so we might have something interesting going on here. So our observation data, I'm still working on processing all these. We have thousands of pictures to go through, so we'll have to stay tuned for this information, but expect this to overlap with our average pollen consumption data. So just some quick conclusions with this. Even though these two pollen types have similar carbohydrate content, they had different levels of protein, which made this an interesting study to move on to. And the bees highly preferred the lower protein brassica pollen over the rose. So one thing there are actually a few things that we still have to consider because there are other factors that may influence a bee's preference and choice, not just protein and carbohydrate content. Perhaps we need to look at the amino acid distribution, the olfactory cues, visual cues. But just moving on to our broader impact, nutrition is very important. Why is this important for you guys? How can we apply this to beekeeping? And really, this is more of there are two different types of science. There's a basic science and there's applied science. And this is more of a basic research because we're doing the science to know and understand what a system is about. Applied science when you're actually seeing if it works in the field. But nutrition is an important driver for colony health today, and there are a lot of synergistic effects of different pesticides, pests, and diseases, which nutrition can help alleviate. And our landscape is changing constantly. That's the whole motivation for me doing the last project where we looked at urban environments because the forage is constantly changing. We may not know exactly what bees are going for. So understanding honeybee nutritional ecology is very important and key to improving overall fitness and moving on to more applied research. So before any applied research, we really have to understand our system. So this is where my research lies. So just some future work is that we definitely want to expand to different pollen types. It was very difficult to get monofloral pollen. I mean, bees like collecting from everything. One thing that we do want to look at is collect the almond pollen and look at that specifically. We also want to look at the amino acid and fatty acid distribution of our pollen types. Recently, just when I was in Denver, a lot of people were talking about how important fatty acids are to honeybees and their preferences. So this is something we're going to analyze. This is one of the first things we're going to do when we get back to College Station this month. And then we also want to look at whether odor will have an effect, the smell of the pollen, and maybe the visual cues. So one thing that we do want to do is to do a proboscis extension response assay. We want to create our own pollen diet to feed them. And eventually the last step would to introduce a stress challenge such as a virus or a pathogen like Nosema and see whether or not that will influence a bee's diet preference for a diet, whether or not they'll actually go for a higher protein diet because they're stressed. So, I mean, that's something we don't know yet. So I have a lot of acknowledgements, but I want to specifically acknowledge Lance Wilson for helping me obtain a lot of this pollen that we used. My advisor, committee members, everyone involved, the Texas Beekeepers Association. With that, I think I have a minute or a minute and a half for questions. So if I don't have time to answer your question, you can find me here. I'll be here the whole time. But yes.
So you showed a graph of 5 days of preferences. The study was over a longer period of time, is that correct?
Yeah, so we had to starve them for a week prior to actually doing the analysis, setting up the pollen dishes. So technically, a study takes 2 weeks officially after you get all the nukes colony setups and all the tent setups. And most of your beekeepers and know how much work it is to set up nukes and set up, I mean, sub colonies. Yes. No, we did this 3 times total. We repeated this experiment 3 times over the summer. What we saw there was the 2 of the repetitions. So we had 4 of them at one time. So that was 8 colonies over 5 days of data. Yeah. I think it depends on the time of the year. Especially winter bees, they need to overwinter, so they. So just overall, I think the context was that the question was, what does longevity mean for the study? And I think that the context of that is nutrition can help supplement that, can help support their health and longevity especially when they're experiencing other pathogens and stressors and the environment. Yeah, well, it depends on the year once again, time. Yeah. All right, I will find you afterwards for your specific question because I think I'm out of time.